Good morning. Welcome, everybody. Thank you for joining us, both everyone in the room and those online. Thanks for taking the time to join us. It's really nice to finally be back to an in-person meeting, so thank you all for coming. We have a full day of talks by NIJ grantees from across our R&D portfolio. You're going to see just a small portion of them. So we have over 150 active grants at any time, and this is just a portion of them. So we also invite you to go to the poster session at 5 o'clock to see more presentations on NIJ-funded work. And of course, throughout the main meeting, there are lots of talks by NIJ-funded researchers. The first 4 talks are going to be from my portfolio, Impression, Pattern, and Trace Evidence. And the first speaker is Steve Lund. Steve is a statistician at NIST, and many of you know he's been involved in lots of applications of statistics for forensic science, and he has taken the lead on this project on forensic footwear comparisons. Steve?
Thank you, Greg. Thank you, everybody, for joining this morning in person and online. Thank you to the organizers and selection committee for the opportunity to talk to you all today about our efforts to bring quantitative evaluation of footwear evidence towards casework application. I'd like to acknowledge the rest of the NIST footwear impression research team, as well as 2 foreign guest research students whose projects at NIST have been helpful in this effort. I'd also like to thank FBI examiners Brian McVicker and Mike Gorn for feedback they've provided throughout this process and for help in collecting case-like impressions for us to work with. Funding for this project is provided by NIJ for the NIST Footwear Impression Research Team, and then from the Statistical Engineering Division at NIST for the foreign guest research students. So under a previous NIJ award, we developed an initial system for footwear impression comparison that we refer to as FIX. At a high level, this system can be thought of as 2 parts, human tasks and computer tasks. The human side of things is typically to interpret image content, which then those interpretations are fed to the computer, which conducts phases of comparison. And the majority of the work for the human comes in interpreting the question impressions, which I'll refer to sometimes as cues. So if this is a crime scene impression, a human is asked to trace out their perception of the outsole pattern seen in that image. And this is used to define a region of interest for the image, as well as used as the basis of comparison when we're comparing against a test impression. So we actually end up comparing the perceived pattern to the test impressions. User is also asked to provide a confidence markup across that region of interest, which is just indicating their perception of the reliability of the different regions or different portions of the region of interest, and is used to weight the different correspondences or discorrespondences when we're computing a similarity metric. So our initial testing for this system came using 3 pairs of shoes that were purchased from the same store at the same time, worn by the same individual. And so with those 6 shoes, here we have 5 test impressions from each of those 6 shoes. Oops. That's what's toggling through here, is just flipping through 5 replicate test impressions from each of those 6 shoes. So that makes 30 total test impressions. We also collected a variety of staged crime scene impressions intended to be a sampling of the types of impressions that may be encountered in casework, and then went to comparing each of the 30 test impressions with each of these staged crime scene impressions. So I'll summarize those results here. So for each of those pictures, the 30 scores are contained in one plot panel. The y-axis here is a similarity measure. And then this is organized so that each of these little subvertical panels is containing the scores from a particular shoe. And then the individual points are the similarity score resulting from comparing a particular test impression with that questioned impression. And for the symbols, the red triangles indicate the similarity scores from test impressions of the actual shoe that made Q. And the black circles are from the other shoes that are used as close non-matches. And the punchline from this plot was for each of the question impressions that we looked at, the similarity scores from the test impressions with the true shoe were higher than the similarity scores from any of the close non-match shoes. So we used this as kind of a proof of principle in producing a second NIJ grant, which sought to apply this to a range of outsole designs and a wider variety of crime scene impressions, and also to try to compare performance between the fix output and human examiner performance. So we reached out to the organizers of the FBI black box study for footwear examiner decisions and asked if they would be willing to share the image sets that they were using to test professional examiners that then we could try to analyze using the fix system. So they were kind enough to send us 270 comparison sets, an example of which is shown here. This is actually figure 2 from their recently published paper. And for each one of these comparison sets, it focuses on a single questioned impression, but it may have multiple image modalities. So in the top left here is an image of the original impression, whereas next to it is a scan from a gel lift of that impression. And sometimes there's a third image as well. Then it also contains 2 different test impressions from a shoe of interest for the comparison set. One is taken from while walking. One was a hand-rolled collection method. And then there's a collection of images of the outsole from the shoe of interest under different lighting angles. So for today's presentation, I'll be talking about results from comparing images of the question impression with the test impressions collected from walking. So of these 270 comparison sets that we received, there were 2 make and models that accounted for just over 2/3 of the comparison sets. The New Balance 878, pictured here on the left, appeared in 49 comparison sets, and the test impressions indicated for each of those that it was a male size 11. And the nice thing about having this appear in so many comparison sets was when we had a question impression, we could compare it not only to the test impression from the shoe of interest for that set, but also the shoe of interest in the 48 other sets. So we had asked to confirm that each of these are in fact a different shoe. So we can actually create a blind lineup of like 49 different shoes of the same make, model, size to aid in our comparison process. Then the most prevalent make model is what we refer to as the boot, shown here on the right, which appeared in just over 133 comparison sets, but from a range of sizes from 9 up to 12. So this not only allowed us to make comparisons with blind lineups of the same make, model, and size, but also same make, model, and nearby sizes. We included up to 1 size difference in either direction. So in total, we made a list of about 12,000 image comparisons that we wanted to make with the FBI data and went about trying to apply the fixed system. And it didn't take long till we realized it was a major headache to be producing manual binary markups for the 580 questioned images provided in this data set. So on the left here is an example of a question impression from a boot, and next to it is the binary markup. And once you're familiar with the tools, this might take about a half hour to produce something like this. But for more intricate designs, such as the Chuck Taylor All-Star, which has these friendly little tick marks all the way around the perimeter, right, this process, instead of taking about a half hour, starts to take multiple hours. And that wasn't even the worst of them, which might be this outsole pattern, which happened to have a very unfriendly question impression where you can see details. So there is information there, but tracing out each one of those individual tick marks for what you perceive didn't leave us feeling very confident about that process. So we sought to investigate 2 pathways of reducing the burden for this manual binary markup. The first was to use technology to reduce the human time required for producing the markup. And the second was to revise our workflow so it doesn't require a full binary markup. And on the technology side of things, we encountered 2 algorithms that started to help us out. The first of those is called superpixel segmentation. So what this does is if you provided an image, it chops it into like jigsaw pieces, trying to find regions of similar colors. And so those regions that resulted from here are shown with yellow. And then we're extracting that yellow region to get the black borders here, which can be used with like a bucket fill operation in Paint or GIMP or Photoshop or whatever your preferred image editing software is, to start producing segmentations of the image. And this worked pretty well when we have large features and we have pretty good contrast. The second tool that we've found useful is we can think of it like an autocomplete segmentation based off of a UNet, which is a convolutional neural network originally developed for biomedical image segmentation. So the way that this works is if you have a question impression, you would pick a few subregions from it and provide manual annotations for those regions, and then use that information as training data for a unit. Once it's been trained, you can give it the entire question impression and ask it to predict what the markup would be like for the entire pattern. And so we saw that there was pretty good correspondence here. It's not perfect, but it looked somewhat reasonable. And so we went forth to try to see, well, what impact would it have if we used the UNet predictions in our comparison process in place of the entirely manual markup. So on the top is a summary of the results that we had seen before, where the human does marks up the entire image, and we saw that for each of the instances, the test impressions from the true shoe produced a higher score than any of the close non-matches. And on the bottom is a repeat of that same process, but substituting instead of the complete manual markup with the prediction from UNet based off of giving a human doing 20% of the image and then using UNet to complete the remaining 80%. And we found that for 7 out of the 11 instances shown here, we see similar behavior. So it substituting in had a gain of efficiency, less time spent marking, and similar performance. But for 4 of the comparisons, we saw a reduction in performance. So this could end up being an iterative process where somebody annotates something, hands it over to UNet, gets back a prediction, and then they inspect it to see if there are any changes that they would like to make. So we're still investigating this. We're optimistic that this can help reduce the burden, but it's not a complete solution, at least not at this point. The second path that we were looking at is to revise the workflow so that it doesn't use a binary markup. So instead of providing a binary markup, we ask the user to mark anchor points, which can be thought of if you have 2 images that have hands in them. And you ask the user to say, could you mark where the tip of the thumb is in both images and the tip of the pinky in both images? And then this tells you how you could reasonably register so that those hand shapes are well aligned as a starting point. So marking anchor points or the removal of binary markups doesn't affect just how we do alignment, but maybe even more so how we were computing our where score. So to understand this, when we were using the binary markups as our basis of comparison from the cues, we know that every time contact is going to be black and non-contact is going to be white, and that edges are going to be a transition between the two. But if we go and look at the underlying actual impression images, we see that there's a much wider variety in how edges, contact, and non-contact appear. Not only between different impressions, but even between different regions of the same impression. So contact in the toe can appear different than contact in the heel, for instance. So when we don't have the binary markup, we have to incorporate greater flexibility into the algorithm to try to capture or represent this information. So we add that flexibility in 2 ways. One is rather than in assessing the entire outsole pattern all at once, we start doing it in patches at a time. And then for a given patch, rather than just using one representation of those pixel intensities, we start to do multiple transformations of that. And that's illustrated here. So in the top left here, this is just a grayscale transformation of the blue box region. Then as we move to the right, that's sequentially applying more and more median blurring. And then underneath is a gradient image for the image above. Gradient is just something that tries to emphasize edges or changes in pixel intensities rather than the original pixel intensities themselves. So the language we would use is this now is like 8 representations of information from this patch, and we don't know which one would be most effective ahead of time, so we actually carry all of them forward to see what works best. For test impressions, it's a little bit simpler because we're dealing with pretty stable information where dark is contact and white is non-contact. So we just have an automated processing for this that leads to a single representation at the end. And that goes from the original test impression. Then we apply an automated segmentation that just labels each pixel as contact or non-contact. And then we convert that into what we refer to as a signed edge distance, which still indicates whether a pixel was contact or non-contact, but also carries the information of how far is that pixel from an edge. And this is useful because pixels that are right at the feature edges often flip between contact and non-contact between replicate test impressions, even from the same shoe. So something right near the edge is less stable than things in the center of a sea of non-contact or a sea of contact. So then now to develop a similarity score, that's where we're doing the comparison between these Q representations and the test impression. So on the left here is assigned edge distance image from one of the test impressions. And then we have our collection of 8 representations from Q. We'll just look first at the top left panel here. And the way the similarity score works is you go to a particular coordinate in the assigned edge distance and record the value of a pixel at that location. So let's say that's represented by the height here. Then you go and find the corresponding location in the question impression representation and find the pixel intensity there. So this pair of pixel intensities can now be represented by a dot there. And then we would go to another location, do the same thing, get another dot. Do this for the entire image. You get a cloud of dots. And the similarity metric is a characterization of the strength of the relationship exhibited by this point cloud. You can think of it like a flexible correlation. Correlations have to be linear, but this doesn't require that the relationship is linear. And in this case, we get a score of 3.18. We then repeat that for each of the representations of Q in turn, so each of them produce kind of a similarity score. And just observationally, we see that for the grayscale representations, applying more blurring got us a stronger similarity score, but on the gradient side, applying no blurring gave us the highest similarity score. So it's not always something that, oh, blurring is good or blurring is bad. It just depends, which is why we keep throwing kind of the kitchen sink of giving these multitude of representations from Q. So once we have a similarity score in hand, we have to decide, like, well, what does this mean? How does this help move a needle one way or the other? And it's important to recognize that a similarity score by itself doesn't mean much. It's not, is this score high or is this score low? But the question we'd really like to know is, does the score from the shoe of interest stand out from the crowd? So if the score from a shoe of interest was much higher than any of the similarity scores we were seeing from a bunch of close non-matches, that may start to increase our confidence that the shoe of interest actually is the true source. And if it doesn't stand out from the crowd, it likely doesn't increase our confidence that the shoe of interest was the true source, and you might be compelled to believe, oh, that means it wasn't the source of Q, which is one explanation, but there are others, such as the shoe of interest could be a really, really complex pattern and that the alignment thing is failing, so we're just not looking at the images in the right way, and so the scores don't stand out. Or that the shoe of interest just doesn't have any distinctive features relative to the other shoes in the lineup. So they all look very much the same, and so there isn't any information there that could distinguish the shoe of interest as the source from Q. Or that Q is just really low quality and it's not capable of reflecting whatever distinctive features that the shoe of interest may have. So rather than jumping to the perception that if it doesn't stand out, that means that it didn't make Q, we want to first check for these other potential explanations. So the way that we do this is taking those scores from the lineup of other close non-matches in the blind lineup. We get a characterization of what scores tend to occur from the other shoes in the lineup. And so this, we're using an empirical thing based off of those multiple replicates, but it also could be something that's modeled to get this distribution. We then apply a second model that tries to answer this question, that if the shoe of interest were actually the true source of Q, how much would we expect its score to stand out from the crowd, given how distinctive it is from the other shoes in the collection, and the quality of Q. So if we had a high-quality question impression and a shoe of interest that had distinctive features, we might expect that its score would stand out quite a bit. So this now would give us maybe a characterization like this green distribution. This then is enough information for us to start answering a question, well, how does a particular score start to change my confidence that a shoe of interest made Q? So if we see a high similarity score, that looks like something that would be likely to occur under the green distribution, but not under the blue distribution. So that would make me start to feel more convinced that the shoe of interest had made Q. If I see something from the center region where both distributions have similar likelihoods, so it's kind of equally likely to see that score under either of the two distributions, that doesn't affect my confidence very much. And if I see a low score that seems much more likely to occur under the blue distribution than the green distribution, that actually starts to make me less convinced that the shoe of interest made Q. Now this was for a particular high quality and distinct shoe of interest. But if I had only moderate quality I wouldn't expect those scores to stand out as much, which means those distributions start to become more and more similar. And the impact of this is that the scores have less capacity to start influencing my confidence one way or the other until you hit to an extreme. If you have a really low quality question impression or a shoe of interest with no distinctive features, then there's nothing that would try to set its scores apart from the crowd. So then regardless of what score you see, it just doesn't have any impact on whether it seems to have made the question impression or not. So we went and applied this approach back with the same Nike data sets here, but now there's no binary markup anywhere. We're just starting with the raw images themselves. And when we compute this and start looking at our performance, we see that we're performing almost as well as we did when we had the complete manual markup available. So the scores here in the red triangles, again, are from test impressions from the shoe that actually made the question impression. The black circles are close non-matches. And there's just a couple of test impression replicates where a close non-match produced a higher score. So before we didn't have any of those. Now we have a couple for this question impression. But we're pretty pleased with the performance given that we didn't have any manual binary markup for this, and we would like to apply this to the FBI data. We do not currently have, we've not unblinded ourselves to what actually is a mate or a non-mate in the FBI, so I can't evaluate the performance of the method in that regard yet. But instead, the information that we do have comes from Table 3 of the black box study paper, which indicates that 107 out of 269 comparison sets were mated, so where the shoe of interest actually did produce the question impression. So this is about 40% of the comparisons were mated. And then if we had a perfect comparison system, if the shoe of interest actually did produce Q, we would like it to produce a higher similarity score than any of the other close non-matches. So if we were to do head-to-head comparisons between scores from the shoe of interest versus the close non-match, the shoe of interest would produce a higher score or win 100% of the time. But then for the other 60% of the cases where it's not the true source, then the shoe of interest is really just one of the random collection of close non-matches being compared. And then it would be a 50-50 coin flip as to whether it produces a higher score or not. So if we blend these two situations, 40% for the top and 60% for the bottom, that gives us a target win percentage of 70%. So we wanted just to go see if that seemed to be true. So on this plot, we're looking at those results. Each panel here corresponds to one of the representations from Q. Grayscale are on the top panels, gradients on the bottom, and each, as we move along, is from progressively applying more and more blurring. On the y-axis is this head-to-head win percentage that for an ideal system, we'd like to see it reach 70%, which is given by the green horizontal line. And the x-axis here is the clarity. So on the left panel, or left side of each panel, are the lowest quality impressions, like where you can hardly even see that there's a shoe impression present. And then on the high end are things that are starting to approach like test impression quality. And this red curve here is just a smoothing curve representing the central trend of these head-to-head win percentages. And we see that at really low qualities, these things start out at about a 50-50 flip. And as we get to really high quality impressions, we're approaching that 70% threshold. So this we view as a really positive result before we actually know on a per comparison basis how well we're doing, but that we're extracting meaningful information in these FBI black box comparisons. And so our remaining steps with this data are we're going to double check our anchor point alignments to make sure we're not starting to compare pinkies with thumbs or anything in those, and just have a second set of eyes look through the clarity assessments, those quality ratings. Then we need to implement the component of the distinctiveness for the Ks, just how unique is any particular test impression among the collection of test impressions. Then we'll unblind ourselves to a portion of the comparison set so that we can fit and apply that likelihood ratio similarity score that we were using as our WHERE score. We'll combine this with our rack and size scores and ultimately compare the results of fixed performance with human performance from the FBI black box study. So thank you very much for your attention, and I think we have time for a few questions.
Thanks, Steve. So we do have time for questions. We can take questions in the room. If you have a question, raise your hand. We have a microphone we can bring to you. Otherwise, you can use that QR code You can text in your questions, both for you in the room and online. Do we have a microphone runner?
Thank you.
Oh, there we go.
Yeah.
Chris first.
Okay. Hey, Steve. I was curious on your log likelihood ratio scale that you were giving. Did I see correctly that the log likelihood was tapping out at about 1?
That was a just cartoon illustration of things. I don't know if I put a 0. I don't know if I put a 1.
I think there was a 1
on the average. For the other results.
Yeah, it should just.
So that's at a per patch level. That was the average log likelihood ratio scale. That's right. So in a 1 inch by 1 inch patch, what's happening with the similarity scores there? That's right.
Okay. And did you accumulate? Do you have a strategy for accumulating across the patches to be able to get an omnibus likelihood ratio?
So, I mean, we could, instead of reporting an average, we can look at a sum, taking like a naive Bayes thing, but we're not trying to, at the end, characterize it through the likelihood ratio as a probabilistic interpretation, but rather as the means to a similarity score that has effective discrimination potential, right? So we eventually want to get to an ordering of the comparison sets so that we can compare the ordering from the algorithm to the ordering provided by the humans. Yep.
Thanks, Steve. So Chris asked my question about the omnibus score, but I do have another one. So I may have missed this. When you are blurring and you go along and you see you have very strong blurring at the very end, how does that affect the edge distance accuracy?
So in our implementation for the signed edge distance, we actually apply that same sequential blurring so that it has the same amount of blurring applied to the binary of k when we're comparing it to the question impression blurring.
Okay, and one more question, if that's okay. The other question I had was when you're doing your anchor points, is there a specific region of the impression that you're covering with the anchor points?
So if one was just doing an anchor point of 2 impressions, we tell them that if you have wider spread anchor points, you get a more stable initialization. But that alignment is only our initial one. After we have a starting point, we then use the similarity metric as an objective function to refine the alignment based off of the actual pixel intensities in the image. But so when we're doing this large collection, we don't want to do 12,000 different sets of anchor point characterizations. Then we kind of create a cumulative library of, okay, we're marking this point A in image A. So maybe for image A we mark A, G, and F, and then in the next one we mark A, F, and Q. And then you use A and F as the pair. And if we don't have at least 2 points, then we go and find an intermediary that you can match image A to B and C to B, and thereby get A to C's alignment. So that's how we're handling it for the 12,000 combinations. Yeah.
All right, we will stop the questions there so we can move on. Thank you, Steve.
Thank you.
Our second speaker of the session is Andrew Paulson. Andrew is a PhD student in the lab of Youngjin Lee at Iowa State University, and he's going to talk about some of the great work on latent print chemistries and time-since deposition. Andrew?
Thank you. Okay, yes, my name is Andrew Paulson, and I'm from Iowa State University. And currently we're looking for oxidation trends that will help us find the time-since deposition of a fingerprint, and we're going to be utilizing something called MALDI, high-resolution mass spectrometry, where MALDI is matrix-assisted laser desorption ionization, and then the MS is mass spectrometry. Okay, so we're all aware that fingerprints can be utilized for suspect identification. However, we can also think about the chemicals that are in a latent or patent fingerprint, and these can be used for evidentiary value as well. The compounds in a fingerprint can be broken down into 2 types of classes. Exogenous compounds, these are the compounds that get on our fingers based on how we interact with our environment, and these can be incriminating compounds or they can be lifestyle information compounds. So the incriminating compounds can be broken down into things like explosives, drugs, firearm residue, and the lifestyle information can be taken from things like DEET or avobenzone. Then we have endogenous compounds, which can further be broken down into things like eccrine compounds. They can come from our sweat glands. These include things like proteins, peptides, amino acids, inorganic salts. And then also sebaceous compounds. These come from our sebaceous glands, things like squalene, triacylglycerols, diacylglycerols, and wax esters. And today we're going to be focusing mainly in on these sebaceous compounds. But because they come from our inner biology, they can tell us some interesting questions about health status, sex determination, physiological age determination, and some other things. So there's a lot of really interesting work going on in the multi-mass spectrometry community answering these types of questions. So especially with the solid alibi, however, there's another question we can try to answer with chemical information, and that is, what is the time since a fingerprint was deposited? We have the who with the ridge detail, we have the what potentially with these exogenous compounds, and then we need that when information. The where is where the crime scene is, and the why, that's a question for another forensic scientist. Okay, so our approach to figure out the time since deposition is based on some work that Dr. Paige Hinners, a recent graduate from the Lee Group, worked on a few years back. And she was utilizing mass spectrometry imaging to analyze the ozonolysis of triacylglycerols over time. So I'm just going to break this down really quick. In her graphical abstract, we have this chemical species, an unsaturated triacylglycerol. Because we're using mass spectrometry imaging, we get ridge detail of these chemicals, allowing us to identify the individual. But then also, we can monitor these over time. Blue means high signal, black means no signal. It's going away over time. So we can potentially monitor that for time since deposition. So here we have the triacylglycerol structure, and we have a carbon-carbon double bond. These carbon-carbon double bonds will react with ambient ozone in the atmosphere. It's about 30 ppb. They'll form these ozonides, and then they'll rearrange. And in our mass spectrometer, we can analyze this aldehyde series, a carboxylic acid series, and a hydroperoxide series. But for now, what we do is that we actually just look at the structure here, the parent structure, degrading over time. So before I jump into the actual results, I want to just quickly mention why mass spectrometry imaging is important to the forensic community. Here we're using matrix-assisted laser desorption ionization mass spectrometry imaging. But how we acquire these, or how we acquire ions to analyze with our mass spectrometer, is by imparting laser energy onto our fingerprint surface and then generating these ions that we can then analyze with our mass spectrometer. And they have the mass to charge ratio that is consistent with these chemical structures. The important thing to know here is that we're analyzing intact molecules without chromatographic separation. And here you can see the laser in this red circle. As we raster that across the surface of our fingerprint, we acquire a chemical profile for each of those laser positions. And then we can take those chemicals for each position and then regenerate these maps. So we get this ridge detail for a chemical image. So here I have a triacylglycerol. Unlike chromatography, the nice thing about mass spectrometry imaging using laser desorption ionization is that we do not dissolve the fingerprint evidence. So the evidence is still intact before and after our analysis. It's semi-destructive, so we do have to desorb things from the surface, but the ridge detail is largely contained. So the work that I'm going to describe today follows this experimental workflow. We acquire our fingerprints. It's pretty preliminary work right now. We acquire the fingerprints, put it on a glass slide, age it in the ambient environment, so we monitor things like ozone, temperature, and humidity. And then we apply our matrix system, desorb the analytes with our laser, and analyze them with our Orbitrap mass spectrometer. And also, before I jump into the data that I'm looking at for new oxidation trends, I want to describe what our current modeling is kind of looking like. So Daphne Patton is going to have a poster today at 5 o'clock if you want to talk to her about the kinetics model. But what she ended up doing was looking at a standard triacylglycerol mixture, aging it at similar conditions to 3 different people's fingerprints. And what we want to see is that the slopes match up. And if you want to get more information about why that is, why we don't really care about the y-axis here, you can talk to Daphne or ask me a question at the end. But we want the slopes to match up. We can see that person 1 matches our modeling quite well for the slope. The person 2 matches decently, but person 3 has a drastically different slope. meaning that we get quicker aging kinetics, and we really want to figure out what is leading to the quicker kinetics in this particular situation. So that is one of the questions I'm trying to address. So what's causing this difference in oxidation rate, and how can we improve this model? So the first step that I thought about was, well, we can just look beyond triacylglycerols. This is a mass spectrum of a fresh fingerprint and an aged fingerprint. And what you can see is that there's a whole lot of chemical information there that we're not probing. So this can be potentially useful for modeling as well. So the goal here, goal number one, is to assess or further understand the chemical complexity of a sebaceous fingerprint aging, and then also look for new aging markers in this chemical complexity. But I also needed a strategy. I can't really look at this and just tell you what the chemical composition is. So the strategy that we utilized, and which was one of the main stories in the publication, was using something called Kendrick mass defect plots to streamline elemental composition annotations. A Kendrick mass defect plot is a scatter plot that uses Kendrick mass defect on the y-axis instead of ion intensity. So here we have our mass to charge ratios, and we have our intensities for each mass to charge ratio. I just take the mass to charge ratios and then plot it with Kendrick mass defect, which is essentially a rescaling of the mass scale of carbon-12 and H2 being 14.000 instead of the traditional IUPAC system based on carbon-12 being 12.000. And I'll get to why that's important in a little bit. But really what we get are these clustering patterns that are informative for heteroatom class assignment. So you can see here that we actually have 3 clusters, and I can rapidly assign elemental compositions. I have an O6 Na series. This is actually the triacylglycerol series. Then when I add a heteroatom class, which is just, or a heteroatom, which is just oxygen, here I go from O6 to O7. I get a shift in my Kendrick mass defect, get a new cluster. From the blue to the orange, what I have is O6 to O8, and then I actually add another sodium. So the oxygens and the sodiums in this case are the heteroatoms, and they cause these massive shifts in my Kendrick mass defect. So just to try to drive this point home a little bit, if I use the IUPAC scale versus the Kendrick mass scale, you can look at the number at the end of each of these mass to charges or Kendrick masses. This is a triacylglycerol structure. It's associated with this dot up here in my Kendrick mass defect plot. If I move over one position, I just add a CH2. The Kendrick mass doesn't change, but the IUPAC mass defect does. And you can see if I add another carbon, I just extend that chain. I move to the right, doesn't change. So these plots are very informative to rapidly assigning elemental compositions. Another pattern that we see going up this diagonal is just adding a carbon-carbon double bond. So that's a little bit of a lesson on how to interpret Kendrick mass defect plots, but really why they're useful is because we go from something like this to something that's well annotated. Oops. So Kendrick plot benefits are that they disperse spectral features by heteroatom class, but it also aligns things called homologous series, which are just series of compounds that differ by CH2 groups in this particular case. Okay, so now I've talked a little bit about Kendrick plots, but how do we apply them to aged fingerprints? As I mentioned, we have these fresh spectra and the aged spectra for sebaceous fingerprint, and it's kind of hard just looking at this to point out key chemical differences. So in order to do this, I just overlaid the Kendrick mass defect plots. So the aged fingerprint Kendrick mass defect plot is a black diamond. A fresh fingerprint Kendrick mass defect plot is the red dot. Any black diamond that doesn't have a red dot in it is unique to the aging process. So these are potential markers that we can utilize to improve our modeling. Okay, so now I actually have the Kendrick mass defect plots, but what are the actual chemical identity of these species? And what I had to do is just look through literature, do a literature survey, and understand that outside of triacylglycerols, we also have things like wax esters, diacylglycerols, fatty acids, and squalene in our fingerprint. And all of these, so long as they're unsaturated, contain a carbon-carbon double bond, can undergo ozonolysis. So I also had to think about all of these structures undergoing ozonolysis and all of them having different carbon chain lengths as well. So I generated an in silico list of potential mass to charge ratios that are associated with all these parent structures, and then all of the ozonolysis fragments as well. And in reality, this was an iterative process, and this is a little bit of a shocking slide, but we get these scatter plots here that are heavily annotated. What I want you to have for a take-home message here is that all the little dots up here in the left-hand corner, those are unannotated series, but it's hard for me to annotate them with a Kendrick mass defect plot because they don't align in these patterns. However, any lipid species do align in patterns, and they're oxidative fragments in our mass spectrum. And the important thing is that they are heavily annotated. I've described a large part of the chemical space here, so we can move forward understanding this chemical space to make our models better. Okay. So another important thing is I'm going to look at the top right-hand side of this plot. This is the triacylglycerol region. It's kind of our ground truth because it's what Dr. Paige Hinders had worked on. And many of the features are defined by ozonolysis. Up here we have the triacylglycerols in this blue box here. And then we don't see too many of the aged fragments yet because that's the fresh fingerprint. We see a little bit of the aldehyde series, but not too much. We see this interesting oxidation called TGE, which I'll get into in a little bit. But for the aged fingerprint, what we can see is that we have the triacylglycerols, we have the aldehyde series, we have the carboxylic acid and hydroperoxide series, and we have a special series unique to the carboxylic acids. So we expect to see all of these patterns, and that's great because our interpretation of the Kendrick mass defect plot is reasonable. However, we see, once again, this new clustering of peaks that is consistent with the O7Na class. Triacylglycerols are O6Na. That means that we're just adding an oxygen to this structure of the triacylglycerols because we're mass shifting from the triacylglycerols up here. So we're adding something without subtracting anything. So we think that this is consistent with the simple addition of a 16 oxygen to triacylglycerol unsaturations, and this was unique to us because we weren't anticipating that with any ozonolysis type fragments. And that is because when you have ozonolysis, you add ozone to our carbon-carbon double bonds, and we fragment that, you must lose this R group here. The R group has carbons, so we have to mass shift down. But in this particular case, we have no mass shifted down. So what we think this is a potential ambient epoxidation mechanism. So we propose this based on some literature, but it could also, and also MSMS data as well, kind of suggested this epoxidation. However, it could also be something like a hydrogen abstraction followed by an insertion of a hydroxyl radical onto the structure as well. Okay. So now Kendrick mass defect plots help us annotate the spectral space and reveal some interesting trends, specifically epoxidation. But we can also do things like remove all the annotations and re-put in the intensity information with these things called Kendrick mass defect bubble plots. So that's what we're going to look at next. We have the fatty acid series, and I'm going to particularly focus in on saturated fatty acids. And this is interesting to me because one would not expect saturated fatty acids to have anything to do with ozonolysis because they do not contain a carbon-carbon double bond. So one would not expect them to be associated with ozonolysis of lipids. However, when we look at our Kendrick mass defect plots in this particular homologous series, which is the fatty acids, fatty acid 10-0 increases over time. So we can just tell that based on the bubble size plot, in the plot increasing. And we see the stark difference, and then when we run our replicates, we can actually see that we get a stark increase in the decanoic acid as well. So what we're attributing this to is actually the carbon-carbon double bond position in the parent structures for most of these, barring squalene. So the fatty acids, triacylglycerols, wax esters, and diacylglycerols may have an omega-10 carbon-carbon double bond position, which just means the carbon-carbon double bond is 10 carbons from the end of the chain. And if you're looking at the mechanism for ozonolysis, this makes quite a lot of sense because when we have ozonolysis of these when we fragment these ozonides, what happens is we get an enrichment of the carboxylic acids, and if they are a carbon-carbon double bond position of omega-10, it would lead to decanoic acid. And actually looking through literature, this is a reasonable hypothesis that this carbon-carbon double bond is here, but we also proved it with something called an on-tissue Paterno-Bucci reaction for carbon-carbon double bond localization, and it suggested that many of the carbon-carbon double bonds are in the omega-10 position. The nice thing is that there's low chemical interference from things like cooking oils or from things like cosmetics, and it also increases our precision at later time points because we get higher sensitivity, and you can think about that as signal dilution. In my mass spectrum, we would have a signal for each of these parent species, but if all of them fragment into one species, it will sum that signal up into one. So that is why I am interested in this decanoic acid as a potential marker. And then it also shows promise to extend our time since deposition modeling, because before we were looking at these parent structures decreasing in signal over time. So we're reaching our limit of detection of our instrument. Now we're monitoring something that's increasing in signal over time, so we're not being cut off by the limit of detection. Okay, so the conclusions here are that Kendrick mass defect plots are useful in the analysis of age-sebaceous fingerprints. We get this informative clustering. Also, we observed epoxidation, which is something that hadn't been observed before, for a potential ambient oxidation mechanism. And then also, decanoic acid is an ozonol-specific aging marker, and one that I think may be powerful as we move forward. So a couple of future directions that we want to take this in is we want to utilize some of the peaks that we identified with our Kendrick mass defect plots to further improve our modeling. So we can look at squalene for fast aging kinetics. We can look at fatty acid 10-0 for long-term aging kinetics. And we can utilize the triacylglycerols that we've been using in the past in order to just keep what we have as a baseline. So we'll try to improve these. We'll incorporate these into kinetics approaches where we get effective rate constants using markers identified in this work along with others. And then also, because we've defined our chemical space a little bit better, we're thinking about doing machine learning approaches, and we've streamlined the data processing because, remember, if you put garbage in a machine learning algorithm, you're going to get garbage out. Now we understand the chemicals that we're putting in a little bit better, so we might have improvements in that realm as well. And then also, we want to apply these new markers to other questions, such as the health-related questions. And with that, I'd like to thank you for your time. We got quite a few people in Dr. Youngjin Lee's group. Trevor had actually just graduated. But Daphne Patton and I are working on this aging study. And if you would like to, please check out her poster. I believe it's at 5 o'clock. It said 5:30 there on accident, but it's at 5 o'clock today. So go ahead and talk to her. All right, thank you for your time, and I'll take any questions.
Hi, good morning. Can you hear me?
Yes.
Yep. I have a question. How stable is this molecule that you're looking at to any external temperature, humidity? Because you mentioned that you're using groomed fingerprint.
Right.
Did you do any studies on how that compound is affected by temperature or levels of humidity and so on?
Humidity hasn't been that much of an effect, but temperature is something that's very important. So what we're doing is we're looking mainly at room conditions. So we spend a lot of money as a society to keep things at a steady temperature. So at relative room temperatures, the kinetics modeling seems to work out quite well. But if you put this at like outside or something, yeah, we're not going to be able to age the fingerprint for sure. But those are definitely important parameters.
Just a follow-up question. So based on if we're trying to deploy this technique in the field, do you think that any transport of any evidence from the crime scene to the crime lab where we would do the analysis would actually affect the stability of those molecules and then misinterpret any possible?
Yeah, so I think commonly what people do, at least I believe so, is that they pick it up and then they put the fingerprint down on a pad and essentially it seals the molecules. We've done studies where we put it in a desiccator or in a closed cabinet and it doesn't actually undergo ozonolysis. So in order to undergo ozonolysis, it has to be open to a large room. Otherwise, in a small closed container, the ozone gets consumed rather rapidly and stops the aging process.
All right. Thank you.
Yeah, no problem.
Thank you. This was an interesting talk. My question actually is a follow-up to the one you just had.
Sure.
Can you comment on the time frame over which this reaction occurs?
Right. Yes. Yeah. So we're right now we've done to about 7 days. So you'd have to have a relatively recent crime for this to be applicable. So it's up to 7 days of aging at the crime scene. But as soon as you capture that fingerprint, it's pretty much frozen in time, so long as you seal it appropriately.
Okay, and just a quick follow-up. So if you have, for example, one of these tags, the triacylglycerides, you've got a double bond there.
Right.
Can you anticipate, for example, that the reaction with ozone would be quantitative or after 24 hours or after 48 hours? I realize that depends on temperature, for example.
Right.
But is that what you expect? In other words, how fast, in terms of days, would one expect full degradation or full reaction at the double bond?
Right. It's about 7 days would be fully done. That's where our limitation right now.
And when it's cold, that...
Right. If it's outside, it would extend that period of time. The kinetics would generally be slower in cold and faster in hot temperatures. Yes.
We have a question from online.
We've received a few questions from our virtual environment. The first is, since fingerprint powder would contaminate the impression, how would you collect the impressions at the crime scene?
That's an awesome question. So Dr. Paige Hinners actually had demonstrated that we can use things like titanium oxide and carbon lifting powder as actual matrices in MALDI. So that's one way that it's actually beneficial to have those there. But we need to extend that. Obviously, this is preliminary work, and we're not using those carbon lifting powders, so we have to really assess what the chemical interferences would be for modeling. But they're actually kind of a benefit for generally MALDI mass spectrometry imaging of fingerprints.
Excellent. What conditions do you use to age the fingerprints?
I think we did this at roughly room temperature, so 22 degrees Celsius, 30 ppb of ozone, and a relative humidity, I think it was like 30% for that, 30 to 40, one of the two. Roughly room conditions.
And one last question from online. Where do you envision this happening in the processing sequence? I assume your technique.
Sure, after sample collection at a crime scene would be where it would be. So.
All right, thank you, Andrew.
Yep.
Our third speaker of this session is Dmitry Kurovsky. He's an assistant professor of biochemistry and biophysics at Texas A&M. Dmitry?
Thank you, Greg. It's a pleasure and honor for me to present at AFS, and many thanks to the organizers for inviting me to present here on this symposium. I'll better put it in my pocket. At Texas A&M, my group puts the effort to advance the forensic analysis of hair. I'm sure in this audience I don't need to remind you and emphasize why the hair is very important, but I want to highlight very quickly that hair is one of the most commonly found evidence at the crime scene. And the forensic analysis of hair can be used to establish a connection between a suspect and a crime scene or demonstrate the absence of these connections. Now, because of this, microscopy is traditionally used to look at the hair, and microscopic analysis of hair can be used to identify whether hair is of human origin or not, and if it's of human origin, what part of the body the hair came from. Is it the scalp hair, facial hair, etc.? and to some extent, what is the race and age of the individual. But obviously, it's not enough, and this problem can be overcome by the utilization of PCR. PCR is very powerful and it's a very accurate technique. However, it is destructive. It is also limited to samples that do have DNA, which is not always the case for hair because the bulb may not be present on the hair sample. But the most substantial limitation of PCR is a laborious nature of this technique and high expenses. In the US, one PCR analysis costs around $25, but in reality of forensic examinations, people wait for months for the PCR results to come. HPLC and HPLC-MS can be used to advance forensic analysis of hair. These very powerful techniques allow for identification of illicit drugs and explosives on hair, but same as PCR, HPLC and HPLC-MS are destructive and also very laborious. But in my opinion, what is largely missing in the forensic analysis of hair is the understanding that half of people around the world color their hair, and they use both permanent and semi-permanent colorants. But what is very interesting is that different colorants have unique composition or unique set of chemicals. Now, obviously, if we can detect and identify these colorants on hair, we can advance the forensic analysis of hair, helping to establish a connection between a suspect and a crime scene or demonstrate the absence of this connection. This to say, I have to emphasize that identification of colorants is a challenging task, primarily due to their low concentrations or low quantities on hair. Therefore, traditional optical techniques such as Raman spectroscopy and fluorescence cannot be used for these purposes. My group proposed the very interesting solution that is based on surface-enhanced Raman. Surface enhancement in Raman spectroscopy comes from noble metal nanostructures that are illuminated by light, and illumination of noble metal nanostructures by light creates so-called localized surface plasmon resonances, or LSPRs, which are coherent oscillations of conductive electrons on the surface of these nanoparticles. The LSPRs, as was demonstrated by my former postdoc advisor, enhance Raman scattering 1,000,000-fold. This 1,000,000-fold enhancement enables single molecule detection. Now, we can use SIRS to probe colorants on hair. All we need to do for this is to take a piece of hair, place it on the glass cover slip. Substrate can be of any kind. It can be aluminum foil, a piece of metal. Then we drop the suspension of nanoparticles. We need only a few microliters to coat the hair, and after this, we illuminate this hair with gold nanoparticles using laser radiation, and we collect scattered photons either using the benchtop confocal Raman microscope or the handheld instrument. So this can be done directly in the crime scene. Now, what actually happens is within a few nanoseconds, gold nanostructures attach or absorb to the surface of hair. And it's very important because the surface of hair possesses the colorants. Now, if the nanoparticles absorb on hair, they come in a close contact with the colorant. And if illuminated by light, they will be able to enhance Raman scattering from the colorants 1,000,000-fold, which enables detection, like I said, of a single molecule of the colorant. This is how it works. If we take a fraction of hair dyed with sky blue and place nanoparticles on them, illuminated by light, within seconds we get this great spectrum that you can see on the left part of the screen. And what is very interesting, first it's an outstanding signal to noise, but also you can take the spectrum as the vibrational fingerprint, the characteristic fingerprint of the colorant. The question is, what do we see? Where the signal is coming from? To answer this question, we looked at the dyeing component of iron blue, which is basic blue 77. It's a semi-permanent colorant. It's just a simple dye. And as you can see, we have a perfect match between the vibrational bands observed in the Ceres spectrum of sky blue and basic blue 77, which demonstrates that what we see is the colorant itself. But it's also important to emphasize that if we don't have nanoparticles on hair, we fail to see the colorant. Again, the nanoparticles are critically important to enhance this signal from very low quantities of the colorant present on hair. And obviously, if the hair is not colored, there will be no vibrational bands. So from here, we can understand that Raman in SIRS can be used to differentiate between colored and uncolored hair, and also detect the vibrational thing of the colorant itself in the hair dyed with semi-permanent colorants. The question to ask, what will happen in the case of the permanent colorant? If the hair is colored by this jet black permanent colorant from iron, and we acquired SIR spectrum from this hair, we get this great spectrum. Now, if we perform the oxidation of phenyldiamines, which are the major building blocks of permanent colorants in a lab, using oxidative agents, we get these high polyaromatic compounds known as Borovsky bases. And if we get a spectrum from these Borovsky bases, again, we observe a perfect match between the vibrational fingerprint of the hair colored with permanent colorants and the dye itself. But in this case, it's not a simple dye, it's a polymers, the set of polymers that are produced by oxidation of phenyldiamines. And here again, every single brand will have slightly different phenyldiamines, primarily for patenting regions, so we get unique vibrational signatures of these colorants. And same as demonstrated for the previous colorant, if we don't have nanoparticles, we have only fluorescence coming, no signal, same as we have no signal if the hair has no colorant on it. And what was very interesting, and we published this result back in 2015, is that different colorants of the same color will have drastically different vibrational fingerprints. Now, if we will have these vibrational fingerprints and utilize them as a library, one can expect that we can enable very accurate identification of a large variety of colorants. This said, we had to demonstrate it. And this was done by a very talented student in my group, Samantha Higgins, that used 33 different colorants, both permanent and semi-permanent and demi-permanent, coming from a variety of different manufacturers like Iron, Vela, etc., and acquired spectra from hair dyed with these colorants. And as you can see on the left part of the screen, every single colorant gave unique vibrational fingerprint that can be used for identification of the colorant itself. But in addition to this visual analysis, my group utilizes chemometric analysis. And using principal least square discriminant analysis, we were able to determine the accuracy of prediction of this spectra to a certain class of the colorants. And as you can see on the table shown on the right part of the screen, in most cases we get 90 to 100% accuracy of identification of the hair colorants. It's quite remarkable, but what is even more interesting is essentially we not only can identify the colorant itself, we can predict the brand where the colorant is coming from. It's very important because every year colorant manufacturers come up with new colorants. So we ask ourselves a question. If the number of colorants is essentially endless, can we track the manufacturer? And here we looked at the vibrational signatures of colorants coming from different manufacturers. And as you can see, out of 700 spectra of iron, 682 were correctly identified as iron. Only 18 were misclassified as Vela, which gives us 97% accuracy of prediction. All 150 of L'Oreal were correctly predicted as L'Oreal, which gives us 100% accuracy of prediction of this brand. And only 8 spectra out of 500 of Vela were misclassified as iron, but the vast majority were correctly predicted, which gives us the 98% accuracy. And for Clariol, all 100 were correctly identified as Clariol. Which tells us that essentially SIRS can be used to identify the manufacturer of the hair colorant. And it's even more exciting that SIRS can also identify the color itself without the need for the forensic examination. And therefore, it can be done entirely automated through automated spectral acquisition. What can be done is using a confocal Raman microscope, spectra can be collected from hair, and through the spectra we can predict the color of the hair, whether it's brown, black, red, blue, etc. And these results are exciting, but I want to remind the audience that to a large extent they were done in the laboratory in a clean, nice environment. But the question to ask is, can this technique be as robustly utilized when it comes to the field examination, when the hair can be contaminated by the substances of biological and non-biological origin? To test this hypothesis, my student colored hair with semi-permanent and permanent colorants, as you can see right here, in an uncontaminated hair. Next, we tested two substances of biological origin, saliva and blood, as well as two substances of non-biological origin, such as dirt and bleach. And what we found is that saliva makes very little, if any, changes to the vibrational signature of both semi-permanent and permanent colorants, telling that it has very little interference, very little, if any, interference with the SIRS analysis of colored hair. The same can be concluded for the dirt. Again, we see vast majority of vibrational bands visible in such spectra. However, what we found, that presence of blood on hair fully obscures the readout of the colorant. We do not see any vibrational bands. The same conclusion can be made about bleach. If hair is bleached, we cannot read out the information about the colorants that were on hair. Well, what we found is that we can clean out blood from hair. All we need to do for this is to rinse hair with large copies of DI water. DI water can be used to remove the blood, but not the colorant. And this hair can be then analyzed using SIRS to identify the colorants, both blue semi-permanent and black permanent colorants. As you can see, the spectra of clean hair match quite well to the spectra of uncontaminated hair. This yet was not possible to do with bleach. Bleach nearly completely erases the vibration, the colorant itself, and therefore it prevents identification of the hair. But what we found is these contaminants of biological and non-biological origin, they cause small changes to the vibrational fingerprints that SIRS identifies. We think it primarily comes from the biological substances present in these contaminants. And if we utilize chemometric analysis, we can predict what contaminant was on hair. As you can see, with quite substantial accuracy, with around 90% accuracy, we can identify whether colored hair was contaminated by saliva or blood or any other out of 4 tested substances of biological and non-biological origin. Another very interesting direction that my group proceeds is the investigation of the effect of environmental factors such as sunlight on the forensic analysis of hair, on the accuracy of SIRS-based analysis of hair. What a talented student did in my group, he colored hair with blue and black permanent and semi-permanent colorants, and then exposed this hair to the sunlight, investigating how sunlight can damage the colorants and consequently alter the accuracy of prediction of these colorants. He collected spectra every 2 weeks over the course of 10 weeks. And what we found is sunlight does bleach the colorants, which is expected because this light-induced photodegradation has been studied for decades by art conservation sciences. One of the most famous study is the Room by Van Gogh, very famous painting. Van Gogh painted it purple, but now we see it light blue because the colorant faded over time. Same we observed here. As you can see, spectra that were acquired in the very beginning of the study, at week 0, are quite intense and strong, and here again they are very characteristic for different colorants. But as time progressed, the intensity of all spectra decreased, but yet didn't fully vanish. And because of this, if we utilize chemometric analysis, we can enable identification of these 4 hair colorants of black permanent, blue permanent, black semi-permanent, and blue semi-permanent quite accurately at all 10 weeks. Here, what is very important is that we have to have these spectroscopic signatures of this light degraded hair, and to enable identification of hair that was exposed to the sunlight. If we utilize vibrational signatures of intact hair, we fail to reach this accuracy, because upon photodegradation, a large number of products of photodegradation appear on hair that contribute to the vibrational signatures that we observe in SIRS. Therefore, it's not only important to develop so-called foundational work, but also it's very important to investigate the changes in the vibrational signatures of the colorants that are taking place upon sunlight, heat, etc., which my group is working at this moment. But the beauty of this work and the beauty of SIRS is it's quantitative. And as you can see here from the ANOVA graphs, SIRS can be used to trace the duration of photodegradation. By analysis of intensity of vibrational bands, we found that permanent and semi-permanent colorants we can trace their degradation and predict how long the colored hair was exposed to the sun. Was it week 1 or week 2, etc. As you can see, intensity of these vibrational bands linearly decreasing as the duration of the sun exposure increases. With all of this, I want to thank my group and primarily Samantha Higgins. She's a fantastically talented student that started this work in my group. Isaac Juarez, very talented researcher, the first generation student at Texas A&M. It's very important for Texas because we have a lot of underrepresented minorities in our region. Thanks to NIJ, he's pursuing his PhD at this moment. Aidan Holman and Mackenzie Stichkovsky, very talented undergrads that are here at this conference and volunteer as the student staff. And also I want to thank NIJ for provided funding, as well as other agencies that help us propelling over the past years. Thank you so much.
Can we get a microphone? Thanks. Hold on, we'll get you the mic so that people online can hear. And a reminder also to submit your questions through the Q&A online and in the room.
Hi, very interesting talk. I have 2 questions. The first question, do you think this method can be used for the dye molecules on the textile fibers? And the second question, you mentioned that this method can distinguish the color dyes on the hair, but let's say the blue dyes have there's different types of blue dyes. Do you think this method can distinguish the blue color dyes, but with different chemical structures?
Right.
Thank you.
So let me start from the last question. Yes, and we have demonstrated this in our laboratory. So the uniqueness of blue dyes from different manufacturers is that different manufacturers have to come with their own cocktail of the colorants to enable its patenting. Therefore, every single manufacturer, despite the color of the dye, will have unique chemical composition. So blue dye from Vella will be different than blue dye from Schwarzkopf. And because of this unique chemical composition, these colorants will give unique vibrational signature in the SIRS spectra that will enable their identification through chemometrics with 100% accuracy, as we demonstrated. So again, the color can be the same, but the dyes present in the colorant will be different. They have to be different for patenting reasons for different manufacturers. Answering your first question, yes, absolutely. In fact, this was done by Francesco Casadia, the very talented researcher at the Art Conservation Institute in Chicago, we collaborated with her on the analysis of colorants in paintings. And crossing the street in Chicago, I came with this idea to utilize this technology for analysis of forensic colorants.
Hi, wonderful presentation. I liked your work discussing contaminants. Have you considered more internal contaminants that people with dyed hair might get keratin treatments or relaxers, and how that will affect the identification of hair dye?
This is definitely an excellent suggestion. Not yet, but we're moving towards that direction. We tested only a small number of contaminants. Right now, ISA Juarez tested a way bigger amount of contaminants, primarily coming from the household substances, substances we can see in the kitchen and bathroom. But your idea is fantastically excellent, and we should look into that.
I have a similar question. So let's say somebody has bleached hair versus really dark black hair. If you put red hair dye on it, it's going to almost just look like a tint on black hair. Or if you put orange or yellow hair dye on black hair, you probably wouldn't even see it. And then also, if somebody has damaged hair, it'll absorb more of the hair dye versus undamaged hair. Do you believe you'd be able to see any of these differences?
It's not I believe, I know. We performed the work in our lab in which we looked at the underlying colorants on hair. And what we've demonstrated that SIRS can be used to detect and identify underlying colorants. If the hair was redyed afterwards with semi-permanent and permanent colorants. And this paper is under review right now. So once published, I'll be glad to share it with you.
We've received a number of questions from our virtual environment. Oh, go ahead, Igor. Go ahead first.
Well, thank you, and thank you, Dmitry, for excellent presentation, which shows further high specificity of vibrational spectroscopy, right? My question is about exposure to sunlight. You mentioned that, in principle, you can determine for how long hair was exposed to sunlight. Don't you need to know two parameters, how much sunlight actually was, maybe there was some cloud on the sky during the exposure. And second, since you are taking quantitative analysis in this case, how do you calibrate, because you didn't know this hair before exposure to sunlight?
Right, two excellent questions. First, we monitor, so the work was done in Texas, and we collected the sunlight intensity parameters over the time course that we performed the SIRS work, and we also reported these parameters in our paper. So we know the sunlight intensity, day duration, etc. But I agree with you completely that perhaps calibration has to be required for other geographic regions, because sun intensity in Texas and the work was done in summer is completely different than, say, sun intensity in New York. So definitely this work has to be continued. Now, the way we calibrated the spectra, we normalized the spectra on CH2 vibrations in this quantitative tracing of the duration of the exposure. But in principle, for identification of the hair colorant, it's not necessarily required. We just take the spectra and we import them directly in the MATLAB.
I had a question on mixtures of dyes. So if you had, let's say in one session, somebody just bought two different colors and mixed them, or maybe they didn't like the coloring and the next week or month they bought a different brand even and dyed their hair again.
Right. I have no answer for the mixtures, but as I already answered, we looked into the underlying hair colorants. In all cases, if semi-permanent was colored by permanent, if permanent was colored by semi-permanent, different variations of colors, etc. And our results demonstrate that Raman, surface-enhanced Raman, can be used to detect and identify underlying colorants, which enables the tracing of the hair dyeing history. But we have done it only with 2 colorants. Of course, the question can be whether if someone dyed it with 3 colorants, can we still identify presence of all 3 and maybe sequential order of the applications? It's something that my group is currently working on.
Okay, and we've received a number of questions online. The first, have you tried comparing the spectra between 2 hair samples rather than identifying the dye? And are the results consistent along the length of hair?
Maybe I will expand that question a little bit. Hair of different individuals with different races, genders, and ages. We have done it, and it's a very interesting set of results. So we looked at the hair that were taken from, I believe, individuals of 5 different races, several different age groups and different genders, and we see that the surface-enhanced Raman spectroscopy can be used to identify the colorants, but these differences, biological differences, should be considered. Now, the hair length isn't matter because we need perhaps 20 microns of hair length to enable identification. So we can work in very small quantities of hair.
Next question. Would we be able to identify hair with other types of nanoparticles besides gold nanoparticles and the differences between the hair dyes?
Yes, absolutely. What matters is so-called LSPR, or absorption band of the nanoparticle suspension. It can be different for different nanoparticles, and the LSPR, or absorption band, will directly depend on the metal, on the size of the nanoparticles, and the dielectric environment. These parameters should be strongly considered relative to excitation wavelengths that is utilized. Gold nanoparticles that we prepare, they're 80 nanometer gold nanoparticles, and if anyone is interested, we can provide you these nanoparticles because we're very happy to disseminate not only the results but also materials that we develop in the laboratory. These nanoparticles are ideal for 785 excitation, 785 laser excitation. But if someone utilizes other excitation sources like 532 green light or 633, we can synthesize nanoparticles that would have absorption band at these laser frequencies to enable the optimal enhancement. Gold nanoparticles are also advantageous because they're relatively stable comparing to silver nanoparticles. Silver nanoparticles can reshape with time. Gold are very stable.
Wonderful. Thank you. And the next question. I didn't see the SIRS spectra for undyed hair, which I might want to compare with the SIRS spectra for dyed hair. Was this viewer missing something?
It was probably the respectful fellow missed that. It was shown in my presentation in a few slides of both semi-permanent and permanent. If the hair is undyed, we do not see any vibrational fingerprints because there is no color and there is nothing for the nanoparticles to enhance. And I'll be glad to follow with this respectful fellow with the papers to demonstrate what I just verbalized in paper-based.
Wonderful, thank you. And last question, this is related, this is actually similar to Igor's question. For the sun exposure work, is time the correct independent variable? Should it be both the product of irradiance and time?
Right, so we consider this parameter of not only of the duration of the exposure that I've demonstrated, but also environmental conditions that we traced over the entire course of the experiment. That was the duration of the sunlight exposure, intensity of the sun, temperature, air temperature, etc. We collected all this information, and it's present in the supplemental information of the paper that was accepted a few weeks ago to Nature Scientific Reports. And I'll be glad to follow up with this information directly to the respectful fellow that asked the question.
All right, thank you, Dmitry. So the last talk of this session is by Brooke Kamrath. Brooke is a professor of forensic sciences at the University of New Haven, and Brooke is going to be talking about automated objective methods for forensic soil examination. Thank you, Brooke.
Thank you very much. Is there a clicker? How do I? Oh, thank you. Let me just advance, and this is okay. Thank you all, and for attending my presentation, and for the NIJ for inviting me here. Normally I save the acknowledgments and thank yous for the end, but I have a lot to talk about, and I don't want to forget to mention the important people who have made this research possible. And in particular, the NIJ has really afforded me the ability to teach a number of students, the 12 of them listed here, about forensic soil analysis, Raman spectroscopy, microscopy, imaging. So coming from a teaching university, that's my products, is the students and their understanding of forensic science and soil analysis. And so I should just sit down now because that's really the most important thing. I also want to thank all of my collaborators as well. The opinions are mine and the authors and not of NIJ, so wanted to make sure that everybody knows that as well. So this is an overview of what I'll be talking about it, but I'm just going to get right to it. So forensic soil analysis is one of the oldest and most traditional forensic traces analyzed. However, I would say over the last 20, 30 years, its use in forensic investigations has really declined for a number of reasons. And so I will take some time today to talk about the value of forensic soil analysis because it's not really being recognized anymore. Hans Gross, who came up with the term criminalistics and is one of the fathers of criminalistics, in his book wrote that dirt on shoes can often tell us more about where the wearer of those shoes had last been than toilsome inquiries. And I'm not going to go through the history, but going from 1893 to just a week ago, ChatGPT also knows the value of forensic soil analysis and wrote the following poem for me. I am not a poet, but soil holds the key to solving mysteries untold in the worlds of forensics. And I think its value is tremendous. One of my favorite cases, which showcases the value of forensic soil analysis, is the Margaret Filbert murder. So in this case, Margaret Filbert was murdered in Bavaria, and they had a suspect, and the suspect, the death had happened, the murder had happened on a Sunday, and the wife of the suspect said that he had worn his Sunday best shoes, and she had just cleaned them the day before. So you don't generally have that great information, but when Georg Popp looked at the shoes, he noticed that there was caked on soil. And he saw that there were different layers, different types of soil. And the layers on the suspect's shoes corresponded to the layers of soil that were important in the case. So first was the suspect's shoes, which contained goose droppings and milky quartz and root fibers and weathered straw. And that was closest to the leather, so it represented a sequential deposit. Next, we had the crime scene soil, which had some red sandstone and angular quartz, and then the murder weapon was found at a castle, and they found soil. He found soil from the castle. So that provided the information, and that when Schlicker was confronted with this, he confessed, and he said, "How did you know all of this?" And the soil had told him. The microscope was really the detective. So when we think about soil, it has a lot of different definitions. I like the definition by Murray and Tedrow in forensic geology that soil is defined as earth material that has been collected accidentally or deliberately and has some association with the matter under investigation. And soil is an incredibly complex material, and this can be overwhelming to think about, but its intrinsic complexity is what gives it its value as forensic evidence. It's the diversity allows us to do provenance determination by looking at these physical variations as well as the differences in the chemical nature of soils. So one of the key features for soil analysis, for forensic soil analysis, is mineral identification. And similar to soil, there's a variety of definitions for what a mineral is. My favorite, and you'll understand why when I go through some of the data, is that it's a naturally occurring inorganic substance having a relatively constant chemical composition and fairly definite physical properties. So there are over 2,000 minerals that exist in the world. The majority of them are rare. About 40 minerals are common in forensic soil analysis. So a forensic soil examiner usually is able to identify those minerals very quickly because you're familiar with the 40, 60, 80 most common minerals. But it is often an unusual mineral, a rare one, that could really provide the most value for associating a question in an unknown sample. Additionally, most soil samples only have between 3 and 5 mineral varieties. So so my mentor, John Refner, taught me about when I evaluate a method, you really want to think about the 3 R's of forensic science. They should be rapid, reliable, and produce a reviewable record. And so when thinking about an ideal method for forensic soil analysis, we would want it to be rapid if we can. Or if not rapid, then automated. So it doesn't necessarily have to be so rapid if it doesn't take a lot of man hours, if I can set it and go potentially. We want it to be reliable, which means we want it to be sensitive, specific, and reproducible. Ideally, we'd want it to be non-destructive, inexpensive, no consumables, or if we do have consumables, there's a big push for everything to be environmentally friendly, also non-carcinogenic, which has been a problem with forensic soil and some methods in the past. Easy to use, but also have not a terrible learning curve, because having a designated operator is not something that's sustainable in a forensic lab. There's been some great tools that have come and labs have used them, but having a designated operator, they haven't, the methods haven't lasted. And then ideally we want it to be comparative and potentially quantitative so that we can do statistical analysis to evaluate its evidentiary significance. And we have a lot of methods for forensic soil analysis, a lot of modern methods, traditional color and texture comparison, particle size distributions, which is what's imaged on the right. Traditional particle size distributions are done with sieves, although we have instruments for that, so you can get much more precise distributions of the particle size using a size analyzer. We also can do density measurements, mineral identification, which I'll mention more of, and as well as the organics and elemental analysis. And we can't forget DNA because that solves every problem. But so in addition to all these tools for soil analysis, we have a lot of different tools for mineral identification. Traditional forensic mineral identification and geology is done with the polarized light microscope, which is one of my favorite tools. But we have a lot of other tools for mineral identification, and my research focuses on Raman spectroscopy. So very briefly, Raman spectroscopy is a vibrational spectroscopy method where you shine monochromatic light on a sample, and it scatters some of that light that's characteristic of the molecular vibration. So it's able to provide a fingerprint for that molecular compound. And it's been successfully used for analyzing and identifying a range of materials. We just heard about dyes, and it's in the geology world, Raman spectroscopy is one of the standards for mineral identification. So my research focuses on combining particle imaging with Raman spectroscopy. Original work that I did with this was using Malvern's Morphology G3, which is a particle imaging system with a Raman spectrometer added to it. And that provided great, valuable information. However, after we were awarded the NIJ grant, we switched to a Hariba instrument, which the Explorer, which is the Raman spectrometer with imaging abilities. So the reason we made the switch is because when evaluating a Raman spectra of minerals, we realized we needed more capabilities for the Raman. So I'll talk more about that in a little bit. But there are multiple instruments available for combining particle information, morphology, particle size distributions with the Raman identification. So what particle correlated Raman spectroscopy does is depicted here. First, we have to prepare your sample. The most important feature for any microscopical analysis is always sample preparation. So first you need to disperse your sample, and then we acquire an optical image that gets converted into a digital image for detection, which can then be analyzed for a variety of different morphological features. And then we can target specific particles based on their morphological information for Raman spectral analysis. So for example, we were able to image in prior research 100,000 particles and then target 3% of those for Raman analysis, which would obviously save a lot of time rather than having to analyze 100,000 particles. And then the spectra can be analyzed for identifying the particles, as well as multivariate statistical analysis. So for particle dispersion, it's the pressure differential is induced by the powder dispersal unit, and you need to make sure you have enough pressure differential to get an even dispersal. So on the left photomicrograph shows a nice even dispersal of particles, not overlapping, whereas when you don't have enough pressure, then you can get agglomeration. Additionally, if you have too much pressure, you can break apart some of the particles. So you need to have the right amount of pressure to disperse onto your microscope slide. Then the particles get imaged, and we target the particles for Raman spectroscopy. Now, there's multiple ways we can do that as well. You can target just the center and do a single spectrum, which is ideal for particle-correlated Raman spectroscopy. Since we are looking to analyze thousands of particles, we can't do a map of 9 over the whole particle. So we really need the single spectrum. And then you image it, and then the instrument, then you do the Raman analysis. And the Raman analysis then can color code the particles based on the Raman signature. So now, instead of just getting particle information about the whole sample, you can get specific morphology, particle size distribution, and other information about each particle type individually. So, for example, we have particle area, and you can see for the particle area of the whole mixture, or of each mineral component individually. We can do particle circularity, the ellipse ratio, and a variety of other components as well. So what have we done so far? So we were awarded the grant in January 2020, and COVID has really thrown a wrench in this research. And we've had some delays for a variety of reasons. So first, I mentioned that the preliminary work was done with the Malvern system, and it was November of 2019 when I was introduced to Hariba's system and Renishaw's system, and I felt that I needed to do my due diligence and evaluate these other instruments. And when I evaluated them, and we realized that the majority of minerals needed more than one laser, and we really needed the best Raman system for identifying the minerals, we switched to the Hariba system. However, This tripled our method development, because now instead of evaluating one laser, we're now evaluating 3 different Raman systems. So essentially tripled our workload for the method development. Additionally, we ordered the system in March 2020, and that's when the world kind of exploded, and we didn't get the instrument until September 2020. So what should have been a 2-month turnaround was 6 months. So I can go through all of the delays, but I don't want to. So what have we done? So we have optimized the method development. So originally we were thinking maybe we wouldn't have to prepare the samples, maybe we could do them neat, but that wasn't the case. You really need to have the washed, dried, and sieved samples in order to get the best information from your samples. And the medium pressure differential really provided the best dispersal without breaking apart the particles. In terms of imaging, you want to collect from the center of the plate. We've collected a lot of soil samples from the Northeast that are ready for analysis. We have some challenges to collecting soil samples in the Northeast. For example, it's very rocky. So with a traditional soil core, we can't get down there. So we've consulted with environmental scientists, and they're saying, oh, bring heavy equipment or other devices. We're like, well, if we can't dig it, then certainly nobody's going to be burying a body in here. So what's the value, right, of digging up rocky soil? So we focus now on topsoil. We've optimized the Raman parameters in a multi-level experimental design, but we need to keep in mind that 1 second, because we're trying to collect Raman spectra from so many particles, we really want to collect it in a small period of time, so 1 second acquisition, which leads to some challenges for Raman identification. So in our method development, we found that Raman can be destructive. We've destroyed particles, so that meant because we want this to be non-destructive, we have to go to a lower laser intensity, which affects the fact that some minerals are weak Raman scatterers. So whereas Raman spectroscopy is a tool for identifying minerals, and it can identify a lot of minerals, it can't do so in 1 second for all of them. So what we're dealing with now is issues with what minerals can we identify, and then that's also overly complicated by other issues. So I mentioned before that that minerals have a relatively constant composition, and even small changes in composition will affect the Raman spectra, which affects the hit quality for spectral identification. So Tim Prusnick from Renishad gave a brilliant talk at Eastern Analytical Symposium in November, where he was using image analysis combined with Raman spectroscopy for geosourcing rutile particles. And the 3 polymorphs of titanium dioxide can be differentiated by the Raman spectra. They can't be differentiated by SEM-EDX. But what he found is some of the particles, this is a single particle. Half is rutile and half is a polymorph of it. So if I'm doing a single point, what's going to happen with this? So we have some serious challenges because we're not dealing with manufactured materials. We're dealing with natural materials with variable composition, and that has affected the hit quality or the spectra themselves, which has a really negative effect on the hit quality when you're doing a library search. So that's where we're at right now, is we can identify the minerals, 75% of them roughly, visually, but the library is not doing so great. So originally we were using the onboard Hariba library, which had about 800 to 900 mineral spectra. We realized that it didn't capture all of the variations, so we had to import the rough database. So the rough database has over 9,000 minerals. Those have to be imported to the library individually, one at a time, which is a lot of work. We also realized we will then figure out what minerals can be reliably identified, and from that suite we'll come up with are match criteria essentially for PCRS in general. So just some of the data. The spectra aren't beautiful, but some of them are. So the titanite is beautiful. It's a 1-second spectrum. We're getting great results. Calcite is noisy, but we can see the peaks. But the library doesn't do a good job with that. But some of the particles are coming out beautiful. But malachite and a bunch of them just don't have a good spectrum. So again, this is where we're at. So I mentioned that about 75% of the minerals can be identified with 2 lasers. So we're doing what we call a double pass. So you're going to have to analyze the sample twice in order to identify the most amount of samples. And conclusions that I have is sample prep is really critical for any mineral identification and for PCRS. There's not a one-size-fits-all method for mineral identification, and so what we need to do is figure out what are those minerals that can be reliably identified with the tool we have, and then determine whether or not that is fit for purpose and able to discriminate the different soils. I have faith that it will provide additional information, and that's what we're working on. So thank you.
Questions?
Thank you, Brooke. Excellent presentation and tough study. 2 questions. First, you showed us that some minerals do not provide good quality spectra. I assume this is due to fluorescence interference?
So it's going to depend. So you have fluorescence for some of them. So that's why some minerals you can identify with one laser, and you have fluorescence with the other. But some just are really weak Raman scatterers and just don't have any identifying peaks.
Okay.
With a 1-second acquisition time. Let me clarify that. So minerals are wonderfully identified by Raman spectroscopy, but often you need a minute, 2 minutes, 10-minute acquisition times. If I'm looking for identifying, I need a short period of time, so I'm not able to identify everything.
Okay, thank you. And the second question is sort of general. I noticed that in the requirements for good forensic tool, you mentioned no consumables. How general it is, and for example, in your case, instead of, yes, you don't need consumables, but your instrument cost over $100,000. You need to maintain this instrument, and you need to do service, right? Annual. So how no consumables is important for forensics?
So I would say that's was my description for the ideal tool. So for the phaser, the dream. So I would say that the consumables makes a difference on what the consumable is. So if it's something like saline or something simple, then that you can order and get, then it's not a big deal. But for example, I do a lot of portable work, and I have portable GCMS, and a little consumable helium is $150. So that is potentially, prohibitive from being useful. I think it'll depend on what the consumable is. But in an ideal world, you just bring it and go, and you don't have to throw anything away.
Thank you.
And we have a question from our virtual audience, or maybe a volunteer situation. Are you interested in audience members making collections for you? I can offer South Texas.
So we're focusing on the Northeast. I am a sand collector. I'm a member of the International Sand Collector Society, so I do collect soil and sand and stuff personally, but for this study is focused on the Northeast to try to really explore the discriminating ability of soils. But it definitely, if this works, it would need to be expanded to other geological regions.
The statistics on that look brutal. Is there any place we can read about that? I know it's too much to ask for a question now.
So the statistics of the original study was published in Forensic Science International, so that's published. I have not published the rest of the stuff that we're doing yet. Still working on it.
So just wanted to clarify here. So based on your soil collections, your sampling, using the traditional methods, are you saying that using just the minerals and visualizing them, you're able to discriminate the soils with 75% accuracy? Was that a correct interpretation?
No.
Okay. So I wasn't clear on what the advantage would be of using your method as opposed to what's currently performed.
So one of the things we are doing is we sent samples to Microtrace, who are doing the traditional soil analysis, and we will compare the results of that analysis to the information you get from PCRS. So the 75% are the number of minerals that can be identified, a rough percentage. So if of the 60 minerals in the most common in the mineral set that we use, roughly 75% of those minerals are capable of identification using our parameters. So it's not about the soils yet. We're still just on the match criteria for the minerals. We're then going to take the mineral identification and use that to form the match criteria for the particle correlated Raman spectroscopy. Now, the advantage of PCRS, ideally, is that not only are we getting the identification of the minerals, but we're getting information about the particle morphology, and that gives information about sourcing and might provide additional differentiation. Also, all of this information is on a large number of particles, so we're going to be able to do beastly statistics on it eventually, but it provides quantitative numbers that could then be used for discrimination. And currently, forensic soil analysis doesn't have the statistics behind it. So it might just be to support the traditional forensic soil analysis, but it automates the mineral identification as well as the morphological information that you get on it from the samples, and that's particularly useful. Thank you.
Thank you to Brooke and all of our speakers. We will take a short break. We reconvene at 10:35 for forensic anthropology and pathology.